Hi guys, my name is Ashish Kumar Jha and I am Assistant Professor at Hislop College, Nagpur. While teaching cell biology to semester 2 students, I was always impressed with the fact that cytoplasmic organelles provide a specialized compartment in which a variety of metabolic activities take place. The generation of metabolic energy is a major activity of all cells, and two cytoplasmic organelles are specifically devoted to energy metabolism and the production of ATP. Mitochondria are responsible for generating most of the useful energy derived from the breakdown of lipids and carbohydrate. And similarly, chloroplasts use energy captured from sunlight to generate both ATP and the reducing power needed to synthesize carbohydrate from carbon dioxide. In this class, we will be studying mitochondria, which play a critical role in the generation of metabolic energy in eukaryotic cells. As told you earlier, they are responsible for most of the useful energy derived from the breakdown of carbohydrates and fatty acids, which is converted to ATP by the process of oxidative phosphorylation. Mitochondria are surrounded by double membrane system, inner membrane and outer membrane. The inner membrane forms numerous folds which extend into the interior of matrix. The, these folds are called cristae. The inner membrane and the outer membrane separated by an intermembrane space. Each of these components play a distinct functional role, with the matrix and inner membrane representing the major working components of the matrix. The mitochondrial genetic system as well as the enzyme responsible for the central reaction of oxidative metabolism. The oxidative breakdown of glucose and fatty acid is the principal source of metabolic energy in animal cell. The initial stage metabolism occurs in cytoplasm, that is glycolysis, where glucose is converted to pyruvate. Pyruvate is then transported into mitochondria where it complete oxidation to carbon dioxide yields the bulk of usable energy, that is ATP, obtained from glucose metabolism. This involves initial oxidation of pyruvate to acetyl coenzyme A, which is broken down to carbon dioxide via citric acid cycle. The oxidation of fatty acids also yields acetyl coenzyme A, which is similar metabolized by the citric acid cycle. The enzyme of the citric acid cycle is located in the matrix of mitochondria and thus are central players in the oxidative breakdown of both carbohydrate and fatty acid. The oxidation of acetyl coenzyme A to carbon dioxide is coupled to the reduction of NAD plus 2 and FAD to NADH and FADH2. Most of the energy derived from the oxidative metabolism is then produced by the process of oxidative phosphorylation. This we are going to discuss in the next topic, which take place inside the mitochondrial membrane. The high energy electrons from NADH and FADH2 are transferred through series of carrier in the membrane to molecular oxygen. The energy derived from this electron transfer reaction is converted to potential energy stored in a proton gradient across the membrane, which is then used to derive ATP synthesis. The inner mitochondrial membrane thus represents the principal site of ATP generation, and this critical role is reflected in its structure. First, its surface area is substantially increased by its folding into cristae. In addition, the inner mitochondrial membrane contains an unusual high percentage of protein which are involved in oxidative phosphorylation as well as the transport of metabolites between the cytosol and mitochondria. Otherwise, the inner membrane is impermeable to most of ions and small molecules, a property critical to maintaining the proton gradient that drives oxidative phosphorylation. In contrast to the inner membrane, the outer membrane is highly permeable to small molecules. This is because it contains protein called porine, which forms channels that allow the free diffusion of molecules of smaller size. Associated with the inner membrane are small particles which have been called elementary particle or subunits of Fernandez-Moran or F0, F1 or ATPase complex. Each particle consists of base piece, stalk and a head piece. The particles are spaced at about 100 angstrom interval. The head piece is 75 to 100 angstrom in diameter and stalk about 50 angstrom. So today we have discussed about